my wonderful KCET as well as PU2 champions. I welcome you all to today's Masterstroke series, where we are going to deal with the chapter Microbes in Human Welfare. Now, this chapter is sort of a blessing for everyone who is tired of reading NCERT, and even after that, they get questions in the question paper which does not seem like it's from NCERT. Because, why did I tell it's a blessing? Because here you get direct questions. So there is no twist and turns out there. Each and every line is very important, and every line is being discussed also. This is a chapter like that, where nothing is being hidden. And my dear children, there is only one challenge. You have to remember the name of the organisms. But let me tell you, if you believe JJ ma'am, everything is possible, right? So yes, and believe in yourself also. Believing in me alone would not help. So let us both do our part, and let us face this chapter, that is Microbes in Human Welfare. Watch this video until the end. And in between, I am going to share you about some free mock test also, which you can get to know. So be there, okay? Let's go ahead. So over here, the first thing that we need to discuss is why are microbes super important in household? That is, in our houses, do you know there are two microbes which are there and they are useful for us? If at all you didn't know, let me tell you. So you might have had curd in your life, right? What is curd? Curd you get from milk. How does milk convert to curd? Basically, if at all you put some bacteria in it, for example, lactic acid bacteria, and the most common of them is known as lactobacillus. This is an example of a group of bacteria, that is lactic acid bacteria. Okay. Now, what does this bacteria do? These bacteria which is there, my dear children, because of the processes they undergo, there comes a position where they would be releasing some enzymes. Okay. Lactic acid to be specific. And lactic acid is being produced because so many other reactions took place before. So anyway, this lactic acid is being produced, and this lactic acid targets on the milk protein. In the milk, you have a protein which is known as casein, my dear children. What do you call it? Casein. Okay. And this casein protein is being partially digested to some form, which is why basically you get lumps of things out there, because it is not fully digested, it is just partially digested. And basically this partially digested forms later converts the milk to what you call as curd. And also remember, while the bacteria does this, it also releases vitamin B12, which is why we say that curd is rich in vitamin B12. Got it? Lactic acid bacteria in the stomach prevents harmful microbes. Yeah, these bacteria, if at all they are in the stomach, they also prevents harmful microbial growth. That is also one very important thing. But as of now, we were talking about household. So just remember at least until this particular point. So what do you have? LAB, lactic acid bacteria, which basically produces some acids that will be coagulating and breaking down, partially digesting the milk protein, which is casein, which is how we get the curd. And also while doing all this drama, they release some very important thing, which is known as vitamin B12, which is super important for our health. Done? Now, the second thing where it is being used, that is the organisms are being used, is for fermentation. You all might have had bread in your life, or if not bread, at least cake. If not cake, at least dosa in Italy and things like that, right? Now, those of you who didn't have breakfast, I hope this video is not released in the morning. If at all it is released in the evening, then let me leave it. Anyways, so yes, so I hope you guys have eaten things, and these things that is there, you might have seen that they are very fluffy. Why are they fluffy? Because over here you are going to use some yeast. Okay, and this yeast is very specific. The name of this yeast is Saccharomyces cerevisiae. Okay, so you're going to use this, and while you use this, they undergo the process of fermentation. You know, there's a process known as fermentation, where basically the sugar which is there, it is going to produce ethanol plus some CO2 and plus some energy. This you might have studied in your 10th grade. If not, then listen to me. So whatever sugar content or carbohydrate content is there, my dear children, that is broken down to ethanol, CO2 as well as H2O. This is a general thing whenever we say fermentation is happening. Now you see this thing that is CO2. This CO2, whenever this gas is being released, that's how you get small pores, and that is the exact reason why things become very fluffy. So basically, bacteria ferment dough. Okay, the dough which is being used, that is fermented with the help of bacteria, and now it produces CO2, and the dough will be what? Puffing up. Yes, and we eat that. Now next thing, baker's yeast. So Saccharomyces cerevisiae, which I was specifically telling, right? So this Saccharomyces cerevisiae is also known as baker's yeast. Why? Because this is used in bread making industries and also for cake making and things like that. Done? Done. So basically we studied the example of curd formation. Second was the example of fermentation of things which may gives us what? Dosa, idli as well as bread. And the example for this is baker's yeast, Saccharomyces cerevisiae, which basically will be producing CO2, and this CO2 whenever it escapes, it gives a puffed experience out there. Okay, now the third category where these organisms are being used is in the drinks as well as foods. For example, you might have heard about toddy. If at all you have visited Kerala, you might have seen toddy shop, toddy shop, toddy shop. What exactly is this toddy? Toddy is basically the fermented palm sap. Okay, the palm, which is the palm trees, no? The fermented palm sap is known as toddy, and for this also you need organisms. Otherwise, without organisms, do you think fermentation can happen? No, Baba, it cannot. Similarly, there are other fermented foods also. For example, fish, soybean, bamboo shoots. Please remember this. This is something that we usually forget because we think it's not important. So there are fermented foods also which people eat, and for this fermentation you need organisms, which is why it is under this particular column. So toddy is there, which you get for fermentation of palm sap. Apart from that, you have fermented food that you obtain from fish, soybean, and also bamboo shoots. Bamboo shoots. Now the last one is cheese production. Bread is done, curd is done. Now it's time for cheese. So my dear children, have you seen Tom and Jerry? If at all you have seen Tom and Jerry, Jerry always wants to have this cheese, but Tom does not allow it to have it. What is this cheese? This is basically the Swiss cheese out there. What is it? Swiss cheese. And in Swiss cheese, my dear children, you see big holes. How do you think these big holes came in? Because from here also the CO2 was released. But who is doing all this drama? It is basically another bacteria whose name is Propionibacterium shermani. So basically Sharmana in Hindi means being shy. Similarly, this is also getting a bit shy because it is releasing CO2, and everyone is seeing that also. So basically Swiss cheese, which is this, is produced by Propionibacterium shermani. Okay, Propionibacterium shermani. It produces CO2, which is why you get large holes out there. Then apart from it, in your NCERT, Roquefort cheese is also mentioned. Now Roquefort cheese, which is there, it is ripened using specific fungi. Now this is a fungi that was a bacteria. This is a fungi that was a bacteria. So for Roquefort cheese, which is a very costly cheese, you need a specific type of fungi. Fungi is responsible for ripening of this particular cheese. Done. So what all did we study? Let us just have a quick go through of it. So we studied there was lactic acid bacteria, for example lactobacillus, my dear children. What does it do? It partially digests milk protein. Partially digest the milk protein out there. And what is the important thing that you have to remember apart from this? Basically they add up vitamin B12. Please do not forget it. Apart from that, we studied in idli, dosa, chutney, money. So listen, idli, dosa, and then you have bread, right? For this, specifically when you talk about bread, we take baker's yeast into picture. Baker's yeast, and baker's yeast is also known as Saccharomyces cerevisiae. Please do not forget it, and please know the spelling of this. Okay, Saccharomyces cerevisiae, which Baba will be doing fermentation, and while it does fermentation, what is going to happen? It releases CO2, which gives a puffy nature. Okay, puffy nature of the dough or the what do you call bread or things like that. Now apart from that, you also use fermented food. Okay, fermented food, and for that again you can use any organism. The name of the organism is not being mentioned over here, but what do you ferment? You can either produce toddy. For toddy production, what do you need? You need organisms, but apart from that you also need palm sap. Okay, the sap of the palm tree. Apart from that, you have some fermented foods such as what? Fish, Baba, and also soybean. And apart from that, bamboo shoots. Now, let's go into cheese production. For cheese production, what do you have? First is there are two types of cheese that you need to remember. One is the Swiss cheese, and the other one is the Roquefort cheese. Now, Roquefort cheese is very costly. So Swiss cheese, which is there, it is being produced with the help of Propionibacterium shermani. And what does it produce? It produces CO2, which is why you have large holes. So please remember Propionibacterium shermani. Apart from that, as I told you, it produces CO2, which produces what? Large holes. Now that was a bacterium, but in Roquefort cheese, for ripening of this cheese, you need what? A fungi. And what do you call this fungi? Actual name of this fungi is Penicillium roqueforti. But you do not need to remember this because this is not given in your NCERT. So just remember fungi is responsible for that. So it's a fungi, here it was a bacteria. Here it was a bacteria. Baker's yeast, yeast is what? Yeast is also a fungus. Yeast is also a fungus, and here you have bacteria. So you can remember like that. Bacteria, fungi. So this is how the flow goes, okay? This is how the flow basically goes. So it will be easy for you to remember in that sense. Cool? Let's go. So this is just, I'm helping you revise also things, but obviously if at all you want, you can pause the video and go for by your own also for revision. Okay? Done. By the way, children, are you liking this video? If at all you're liking this video, click on the like button right now, because I feel really happy when I see likes and comments coming up there saying, ma'am, ma'am, you teach good and things like that. Obviously, I also need some motivation, right? So if at all you think that you're understanding things well, do not forget to hit the like button. And I was just joking. Please share it with your friends also. You did not comment and all. Just share it with your friends who are in need, if at all you think they are in need. Okay, now let's go ahead. Microbes in industrial product. So now in industries we see microbes are being used, and the first type of industry which is very, very famous is beverage industry. Is what? Beverage industry. Okay, so what happens in the industry? In this industry, so used to synthesize valuable products, for example, beverages as antibiotics, okay, grown in large fermenters for large scale production. Yes, so all these beverages and things, apart from that, even antibiotics, whatever is there in industries, my dear children, they are produced in large fermenters because you need a bulk of things to be produced. And here the microbes which are being used, let's talk about that. Here it is brewer's yeast. Okay, is this yeast different? No, Baba, it is the same yeast, that is Saccharomyces cerevisiae. But since you are using in brewery industry, you can also call this as brewer's yeast. So it can be known as baker's yeast, it can be known as brewer's yeast, depending on for what it is being used, okay? So it ferments malted cereals and fruit juices, produces ethanol. Listen, I told you at the end of fermentation, what all do you get? You get ethanol, you get CO2, and you get ATP, which is why we say the idli, dosa, batter and all, if we keep for some fermentation, it gives a very, a little bit of sour taste out there. Why is this sour taste coming up? Because of the ethanol. Little amount of alcohol is being produced. If at all you keep it for a very long time, more will be produced. But anyways, okay, so our goal over here is to basically produce alcohol, and ethanol is basically that. Yes, so Saccharomyces cerevisiae undergo fermentation helps in the fermentation process for making up of what? Ethanol. But what is it fermenting, my dear children? You should know that, and this is a question that has been coming, and which is why I'm telling you it's important. So they usually ferment malted cereals. Malted cereals means these cereals, my dear children, they were about to undergo germination and produce a new plant out there, but it is being stopped. Just when it was about to, it is being stopped. So this process is known as malting. So malted cereals are being taken. Apart from that, fruit juices are also being taken. Fruit juices, malted cereals and fruit juices are being fermented to produce what? Ethanol, which is an alcohol. Now fermentation pathway, as I said, converts sugar to ethanol and CO2, etc., etc. So you know that, I already spoke about that, right? Now types of alcoholic drinks, this is important. So there are two types of drinks. One is the one which you get without distillation. Without distillation means what? So usually, suppose there is an alcohol over here. Suppose whatever water content is there, it is made to be evaporated. So what do you get? You get alcohol. And what form of alcohol? Concentrated form of alcohol, because the water is being completely, or basically almost removed, not completely, but most of the percentage removed, my dear children, through evaporation process or through another process. This is known as distillation. It's known as distillation whenever you do that. So at the end of it, what do you get? You get concentrated alcohol. So the things which makes people go like this, that means that probably in that the alcohol concentration could be more. That means they might have underwent the process of distillation. So what are the things that gives the kick, in the sudden kick out there? Obviously the strong drinks, for example, you talk about whiskey, brandy, rum, all of it. But there are certain things which do not need the process of distillation. You do not do distillation, the water has not been evaporated, like, oh my god, everything went. Anyways, the water has not been evaporated, which means that it is still diluted. It is not very concentrated alcohol that we are drinking. It is still very diluted, right? So this is something that does not give very fast kick, and they will obviously have water as well as alcohol in enough quantities. So example, you have wine and beer. You have wine and beer. So whiskey, brandy, rum, all of it has a higher concentration of what? Alcohol. Has a higher concentration of alcohol. But when you compare with wine and beer, obviously they will be having lower concentration of alcohol. It's not like you are having concentrated alcohol only. Okay, when it undergoes distillation, it's not like you are having concentrated alcohol. Everything will be, what do you call, fired up. It's not like that. So there will be amount of water definitely being there also, but not too much. That is the thing. That is the logic. So without distillation, that means less concentration. Here it will be wine and beer, and with distillation, that means whiskey, brandy and rum, because there will be more concentration of the alcohol content out there. Okay, fine. So what did we get at the end product of this? Microbes in industrial products. The first industry is that brewery is the what? The brewerage industry. Over here you are going to use a different type of yeast. What do you call this type of yeast as? The same type of yeast only, not different. It is known as brewer's yeast. Saccharomyces cerevisiae itself. What does it do? It undergoes fermentation. Fermentation of what? Fermentation of malted cereals as well as fruit juices. To produce what, ma'am? It is to produce what? The ethanol as well as CO2 and things like that, especially ethanol. Okay. Now, there are two types of drinks. One, without distillation. Without distillation means evaporation is not happening much. Concentration is not there much. It's diluted stage, right? Little amount of water is still being missed. You call this as without distillation, and basically the products of it is wine as well as beer. Whereas with distillation means the water content is being blown away and you have more concentrated form of alcohol up there. This is seen in rum, brandy as well as whiskey. Got it? This is a very famous question. Yes. Now we move on to the next industry, which is the industry where chemicals are being produced, where enzymes are being produced, and where bioactive molecules are being produced. Okay, so we'll go one by one. We'll talk about chemicals first, then we'll talk about enzymes, and then we'll talk about bioactive molecules, right? So in your model question paper, the question has come from this area. We can expect something to come around from here. So make sure you know about this thing really, really well. Okay. So chemicals in general are what? Citric acid, acetic acid, butyric acid, lactic acid and ethanol. So let's undo the things that we have already done. As in, let's not undo the things, let's mark the things that we have already done. We have already studied that how do we get lactic acid? With the help of lactobacillus, that is a bacteria, or you call them lactic acid bacteria in general. But if at all you want to give a specific name for a single bacteria, it is lactobacillus also. Fine. Ethanol also we studied, we get it with the help of Saccharomyces cerevisiae, if at all it undergoes fermentation process. So yeast is what, my dear children? Yeast is a fungus and lactobacillus is basically a bacterium. Please know about that. Now we have three new guys up over here, and three new guys. After that, two of them are very, very easy to remember. Acetic acid. Acetic acid you get from Acetobacter aceti. Now acetic acid, Acetobacter aceti has a commonness in their name, right? It sounds similar. Acetic acid and Acetobacter aceti. Basically, acetic acid you get from Acetobacter aceti. What is it? It's a bacterium. It's a bacterium. What about butyric acid? Butyric acid you get from Clostridium butylicum. Clostridium butylicum. Again, butyric acid, Clostridium butylicum. Buta has a similarity out there. So this is also easy to remember. You will be able to match the following very easily, right? Now, one thing is there which does not have any association with the name. Citric acid. Citric acid is produced from Aspergillus niger. What? Aspergillus niger. And Aspergillus niger, my dear children, is a fungus. Is what? A fungus. So you saw ethanol is produced with the help of fungus, as well as citric acid is produced with the help of fungus. Just all of them, my dear children, over here are produced with the help of bacterium. Now obviously you will not study in this way. We need to have a tougher way to study, right? For that, what do? We do recalling. We do recalling. So let's start with the organism that is fungus, and what does fungus produce? Okay, so the first thing that we have is ethanol, and ethanol is produced by what? Saccharomyces cerevisiae. Done. First thing is being done. Second, fungus. Okay, we're talking about fungi. Second is citric acid. Guys, citric acid is produced by what? The one that does not have any name association. It does not sound similar. It is going to be what? Any idea? Citric acid? It is Aspergillus niger. Aspergillus niger. This is the next fungus that we have that produces citric acid. So the fungi thing is being done. Now we talk about the bacteria. And the easiest bacteria that we already know is what? Basically, first of all, the product, and that is lactic acid. It is produced by lactobacillus. Right? Now we have acetic acid. And we have one more thing, that is what? Butyric acid. Butyric acid. Come on, recall. Acetic acid is produced by what? It's a bacterium, has a similar name. Acetic acid is produced by what? Acetobacter aceti. What do you call it? Acetobacter aceti. Okay, fine. Now let's go ahead. We have the next thing, that is butyric acid. Butyric acid is produced by Clostridium butylicum. Clostridium butylicum. Please know that. All of this is super important. So this is why am I writing over here? For you to recall. It's not like it's for you to check the spelling and things like that. It's just for you to recall it. Okay. Citric acid, Aspergillus niger. Acetic acid, Acetobacter aceti. That is the bacterium. Butyric acid, Clostridium butylicum. Lactic acid, Lactobacillus. Ethanol, Saccharomyces cerevisiae. Done. So that is the chemicals that we have totally done. How many chemicals are there in total? 5 of them were there. Now we talk about the enzymes. In enzymes, you have lipases, pectinases, proteases, and streptokinase. So lipases is the first one. Lipase, my dear children, by the name you get to understand that it is going to break down lipids. Lipids as in basically we're talking about the fat. For example, on your dress some oil spot came up. What do? You use detergents, right, to remove this. What does a detergent have? Detergents will be having some enzymes basically, that is lipases. These lipases are going to target and going to break down the oil out there. That is how it is going to remove the stuff. So used in detergents to remove oily stains. Okay. Now we have pectinases and proteinases. Where do you use that? So, pectin and proteins. Pectin is basically a part of the cell wall of the plant cell, and proteases is for breaking down the proteins. So, basically you might have seen the bottled fruit juices out there. So, just clarify it, so to make it clear, otherwise it will be coming up, right? So, to clarify it, we use pectinases because it is going to break down the cell wall, as well as proteases that breaks down the proteins. So, to clarify the bottled fruit juices that we have, we need pectinases and proteases. Now, the third enzyme is super important. Okay, and that is streptokinase. Streptokinase. Just suppose a person is having heart attack. Okay, heart attack comes because of myocardial infarction. So, myocardial infarction causes heart attack, my dear children, and basically in this particular condition, it happens because there is a clot in the body. There is a clot in the body. And we have to remove this clot such that the blood flow can easily happen, which is why we would need what? Clot busters. We need someone to burst down the clot, and that is known as clot buster. Who does that? This particular enzyme. Streptokinase is an enzyme that acts like a clot buster. Ma'am, where is it being produced from? It is produced from a bacteria whose name is Streptococcus. Its name is what? Streptococcus. Please remember, super important. So streptokinase you get from Streptococcus, which is basically helping in busting down the clots are there, and this is very important in people who have suffered myocardial infarction leading to heart attack. Okay, so all of this are what? Enzymes out there. So how many enzymes do you have, my dear children? You have the first enzyme, that is lipases. What do? They break down the lipids. Where are they found? In the detergents. Why? Because we use detergents to wash down the oily stains. So to remove the stain out there, we need lipases, which will break down the oily thing, that is a fat or the lipid. Okay, going ahead, apart from that, what do you have? You have pectinases and protease. Pectinase is a compound that is found in the cell wall of the plant cell, and also protease is the compound that is found, protease is an enzyme that is basically used for breaking down the protein molecules out there. So this is being used up in bottled fruit juices out there, so that there is no clumps being formed, and it is very clarified. Okay, it has a good visibility of how it looks. Okay, the next thing that we have is the last one, and that's the most important one among all of them, and that is streptokinase. Streptokinase, my dear children, you get it from Streptococcus. Streptococcus. And what does it do? It's a clot buster. It is a clot buster which is used during myocardial infarction. Myocardial infarction. Please remember this. Super important. These are what? Enzymes. Okay? Yes. Now we come up to the last one, that is bioactive molecules. Again, super important. Bioactive molecules. We have two bioactive molecules. One is cyclosporin A and the other one is statins. Cyclosporin A is from a fungi whose name is Trichoderma polysporum. Trichoderma polysporum. Okay, what does it do? It is an immunosuppressant. Cyclosporin A, which is a compound that I get from the fungi Trichoderma polysporum, is an immunosuppressive agent. For example, I took the kidney from you because my kidneys were not good. But my body will react to it because it's foreign substance that has been there. But I do not want my body to react to it. I want my body to welcome this organ up there because it is letting me live, right? So in that particular case, I want to make sure that my immune system is not too active, because if it is, it might probably kill the foreign substance that is the new kidney up there. So in this case, what do you take? You take the help of immunosuppressants. So cyclosporin A is one such immunosuppressant out there which is used in organ transplantation, and also you get it from the fungi Trichoderma polysporum. This is a fungi. This is a fungi. After a long time, we're talking about a fungi over here. Similarly, statin, which is there. So statin, the name is very common with stabilizing. Statin stabilizes. Statin stabilizes what? The blood cholesterol level. Okay, statin stabilizes the blood cholesterol level, or actually it is known as a blood cholesterol lowering agent. Okay, it lowers the cholesterol by inhibiting its synthesis. So statin, you get it from Monascus purpureus. You get it from Monascus purpureus, and what does it do? It lowers the cholesterol by inhibiting its synthesis. How does it lower? It does not allow it to be synthesized itself. So statin, Monascus purpureus, it lowers the cholesterol by inhibiting its synthesis. Done. Now, these are the bioactive molecules. So over here again, just a second. Bioactive molecules, how many do we have? We have cyclosporin A and we have statin. Right? Cyclosporin A is what? Is basically an immunosuppressant. And statin is basically what? It is a blood cholesterol, lowering of blood cholesterol. Done. Now, listen to me very carefully. Where do you get it from? You get it from a fungus whose name is Trichoderma polysporum. And where do you get it from? You get it from Monascus purpureus. Monascus purpureus. Ma'am, how do you remember the names? By teaching you guys. So if at all you're facing difficulty in remembering all this name, teach it to someone else. Think that someone else is there in front of you and start teaching. Anyway, you'll remember things. And it's not like I do not forget. I also forget. But we have to keep on revising to keep it really strong in our head. Got it? So that is the story of bioactive molecules. So we have studied the chemicals, we have studied the enzymes. In chemicals, citric acid, acetic acid, butyric acid, lactic acid, and ethanol is there. Two things you get from fungi, that is Aspergillus niger and that is Saccharomyces cerevisiae. Aspergillus niger gives you citric acid and Saccharomyces cerevisiae gives you ethanol. Apart from that, we spoke about enzyme, lipases, pectinases, proteases, and also streptokinase. Streptokinase you get from Streptococcus, which is a bacteria, and this particular streptokinase is a clot buster. Okay, used during myocardial infarction leading to heart attack. Bioactive molecules, you have cyclosporin A that you get from Trichoderma polysporum, which is an immunosuppressive agent, and statin, which is a thing that is responsible for lowering of blood cholesterol. You get that from Monascus purpureus, and it inhibits the synthesis of cholesterol. Done. Now, the next industry is antibiotic industry. The next industry is antibiotic industry, and with that we are going to finish the microbes in industrial product. So if at all in your PU2 you get to a question that is basically explain the role of microbes in industrial product, you have to explain all of it. That is the first one onwards, brewery, industry, then this and then this. Usually they're not long questions. They ask you MCQs on the basis of this. And sometimes they can just give you antibiotics, explain that, or chemicals, things, and either one of them. Okay, explain all this thing together. So that can be coming in. So that's the reason that I'm telling you it's important. You need to know about it. Any part of this chapter can be coming in, but yes, it is more predictable. Cool. Yes, predictable as in something apart from what I'm teaching you has no chance to come. That surety I have. You get it? That's why I'm telling you it's more predictable. Cool. Now, antibiotics, we're talking about antibiotics. Antibiotics means what? Anti means against. Against, biotic means what? Life. So, antibiotics are something that are taken against life. It kills, inhibits microbes. Yes, but definitely it is super useful for us. It is produced by microbes used to treat bacterial infection. Okay, now let's talk about how was the first antibiotic being made. It's a very funny story. So there was a person whose name was Alexander Fleming. So suppose I am Alexander Fleming. So I am working on my famous organism out there which is known as Staphylococci. Oh, just a second. Okay, I was working on Staphylococci bacteria. And I was looking at its growth and things like that, and I felt sleepy. I went to sleep without doing anything about the dish over here. I didn't wash this. The next morning I woke up, I was like, oh my God, what did I do? The bacteria might have grown so much, it might be a huge trouble for me now. Now when I entered the lab, I saw that this bacteria was not able to grow, which is something very unique, which he didn't find common. So Staphylococci, it's not Streptococcus. Do not go and write Streptococcus and come. Streptococcus, Streptokinase. Okay, this is Staphylococci. So he was there right over here. He was thinking, what's the reason? Why didn't it grow big? What's the reason? So he came to a conclusion that something is being produced here which is like a mold. This mold is known as Penicillium notatum. Penicillium notatum. This mold is there, and probably because of this mold that guy is not growing up. But what could probably be the reason? Then he researched about it and got to know that the mold is releasing something which is not allowing this organism to grow. And that substance which was there is an antibiotic. The one that does not allow the other organism, that is the the other microorganisms to grow. It's just known as antibiotic, especially when you're talking about bacteria. It does not allow the other bacteria to grow over here. So this is an antibiotic that was being released, antibiotic. And what was the name of this antibiotic? It was produced from Penicillium notatum, and the name of this antibiotic was penicillin. It is what? Penicillin. So penicillin was the first antibiotic that was being produced. The first antibiotic discovered by Alexander Fleming in the year 1928. Please remember, 1928. He found that mold, Penicillium notatum, inhibiting Staphylococci growth, later developed by Ernst Chain and Howard Florey. So it's not just Alexander Fleming. Okay, he definitely discovered that first. So he was the first person to do that. But apart from that, Ernst Chain as well as Howard Florey came into the picture and got a better understanding of this as well. Okay, so Ernst Chain and Howard Florey, do not please forget them. And you know what? Three of them, Fleming, Chain and Florey, three of them got the Nobel Prize for this. You know, you could treat a lot of diseases that were there because you have potentially made something that could kill bacteria, and bacteria used to cause a lot of trouble before, right? So yes, the first antibiotic was being celebrated. So which is why Alexander Fleming, Chain as well as Florey got their first Nobel Prize in the year 1945. So 1928 he discovered, 1945 they got the prize. Before that, you need to understand that it was used in World War II to treat wounded soldiers. So this particular antibiotic was used in World War II to treat what? The wounded soldiers who were out there. So please know that also, World War II. Cool. So what do we have in stock? My dear children, always remember, penicillin is the first antibiotic that you got. You got it with the help of Alexander Fleming's experiment. He was working on Staphylococci. Then he saw that it was not able to grow because of the formation of a mold. That is Penicillium notatum. That is a fungi. It's a fungi. Okay, when I say mold, it's a fungi. This Penicillium notatum which is there is producing what? An antibiotic, that is penicillin, that does not allow the growth. Later, it was developed much more by Ernst Chain as well as Howard Florey, and three of them together got their Nobel Prize. Apart from it, in World War II also this was particularly being used. Done. That's all. This is microbes in industrial production of antibiotics. And with this, we end the industrial part out there right away. The next thing that we are going to discuss is about the sewage treatment, which is yet another very important topic. Before I go ahead with that, everyone, if at all you scroll down the lecture, basically you will be able to find something like this. free KCET PCMB mock test PDF. So this is a PCMB, physics, chemistry, mathematics, biology mock test. Now don't say you guys are not like that. You will anyway need biology, right? Some people say we do not need biology because anyway we are not giving KCET for bio. So yes, that hurts me, but yes, it's okay. So my dear children, you can take the best use of it. It's for free. So click on the link. Once you click on the link, your mobile number will be asked. You can give the mobile number. Post that, you will be allowed to get the PDF out there. So this PDF will be received. All the subjects are there. Okay, do not be worried. All the subjects are being clubbed up together. So yes, you can make the best use of it. Yes, so that's a mock test. Please give the mock test. It could really help you with preparation if at all you are someone who is ready to ace KCET examination. Yeah, so just have a go, have a look at it. Probably start solving some questions. You will get to know where are you liking, where you're not. Yeah, cool. Now, everyone who is out there, we have also launched ABHYAS KCET test series. I hope you guys are aware of it. Yes. So what do you get over here? 35 KCET mock test. So not just one, it's 35 KCET mock test. Apart from that, you have chapter-wise PYQs. This is something that we have added up this year, thinking that if at all you are someone who is preparing for your PU2 boards, it might be difficult for you to attend the mock test as of now. So yes, you can utilize your time best in solving PYQs because it won't take much of your time. 60 minutes, 60 questions. So if at all you don't want to, solve all the 60 questions, at least 15 questions at one go will be enough. And these MCQs, my dear children, will definitely help you in your PU2 MCQ preparation as well. Okay, and all of it is under perfect timing, so it gives an idea about your speed and accuracy also. The price for this is rupees 1499, and don't worry about how is this particular test series, because we have proof. We have got ranks like 171, 443, 693, and there is a whole list up there. So yes, thousands of children trusted us. They got the best use of it. You can also do that. If at all you buy some book for all this thing, it costs almost similar, right, in MTG and things like that. So yes, it's a very decent price. If at all you're interested, you can take a look and go buy. In the description section, there will be a link for that also. Okay, now we talk about the sewage treatment. Okay, so how does it go? There are two types of treatment, primary treatment as well as secondary treatment, and we'll go one by one for both of them. Okay. What happens in primary treatment? We do physical removal of things, very easy thing. So it removes large and the small particles from the sewage. There is two processes involved in primary treatment. One is filtration, another one is sedimentation. Filtration means whatever things can be floating around at the top, my dear children, you can remove those floating debris. That is known as filtration. Sedimentation means whichever thing is very, heavy, obviously they will be settling down. So you can remove through that. So it removes grit, soil as well as small pebbles which are heavy. Now basically these things which are there, the settled solids which are there, everything which are being floating around, all of this, what do you call this as? The primary sludge. Do we need it? No, we need to remove it. Whatever is left behind is basically the fluid which is up there, and this is known as the primary effluent. What do you call it? Primary effluent. So I will talk about the story one by one. Make sure you listen to me very carefully. Okay, and just give me a second. I am looking out for something. Okay, so over here, this is a place where we are going to deal with things. So this is our primary treatment. What did we do? We did two things. One was filtration. Whatever big things, those were removed. And one was sedimentation. So this is sedimentation, through which we are removing things, and this is filtration, through which we are removing things. Two processes done. And now whatever is left behind is known as the primary effluent. And what is this tank where everything is happening? You need to know the name of the tanks also. So primary treatment is being done in a settling tank. In a settling tank. Ma'am, is this important? Yes. So see, settling tank, everything is getting settled down here. Everything which is floating down, and whatever is there, if over here, that is known as primary effluent, right? This primary effluent which is there, we are going to take it for the next treatment, that is known as secondary treatment. And what do? For secondary treatment, you are going to put this effluent in a tank which is known as aeration tank. So from settling tank, you are now shifting to aeration tank. Why? Because here you are going to provide a lot of air. Yes, I am actually correct. So you might be thinking that I am joking, but I am actually not. So over here, this primary effluent which is there, it will contain high amount of organic matter, right? Things have to be broken down. There are a lot of things that has to be broken down and have to be made clear. This is known as organic matter. And to break down this organic matter, what are we adding up over here? We are adding some bacteria up which is known as the aerobic bacteria, which is going to be the hero or heroine over here. The main cast is what? The aerobic bacteria. Aerobic bacteria we're putting in aeration tank. Why? Because the aerobic bacteria will definitely need the air. To do what? To basically break down the organic matter. And it does not do it alone. It also needs the help of some fungal filaments. Some fungal filaments. So aerobic bacteria plus some fungal filaments are going to make mesh-like structure out there. This is a fungal filament that I am making, okay? They form a mesh-like structure out there which you call as flux. Which you call as what? Flux. So what is flux? This is a very important question. Flux and BOD is a very important thing. What is flux? Flux is nothing. The aerobic bacteria plus the fungal filaments together, you call this as flux. What is the importance of flux? Flux is something that is going to break down the organic matter. It is going to break down the organic matter out there. And while it happens, my dear children, I told you there is what? A need of air. And by air, I mean oxygen. Aerobic bacteria needs oxygen to do this process. So if at all you think, okay, if at all organic matter is more, that means if I say that waste is more, obviously there will be need of so many aerobic bacteria. And if there is aerobic bacteria which is in more amount, my dear children, that means the need of oxygen is also more. This demand for oxygen is known as biochemical oxygen demand. And in this case, where the amount of waste is way too much, since aerobic bacteria will also be way too much, and since they will be needing too much of oxygen to break them down, I'll say the BOD, biochemical oxygen demand, is going to be more in the polluted water bodies. On the other hand, if at all, okay, if at all the organic matter, that is waste, is less, that means aerobic bacteria will be less because there are no too many things to break down, right? Which is why the oxygen demand is also going to be less. That means the biochemical oxygen demand would be also less. Got it? So less pollution means biochemical oxygen demand will be less. More pollution means biochemical oxygen demand will be more. The demand is more. Why? Because more amount of oxygen is being taken up by the aerobic bacteria to break this organic molecules. And it's not just aerobic bacteria, it's basically the fungal filaments together. You call this as flux. And where is this thing happening? In the aeration tank, because you need a lot of air. So first was in the settling tank, then aeration tank, and now we are going to shift it back. Shift this thing back into the next tank out there, which is again the settling tank. So we started with settling tank. We are again going to put it in another settling tank. So in this settling tank, what is going to happen, my dear children? Everything which was up over here, we are going to shift it right over here. And you will be seeing something like this settling down. Whenever you want settling down, that's when we use settling tank, right? And you have this. What is this, my dear children? You can clearly understand I have made the aerobic bacteria plus the what? The fungal filament. So can I say it's the flux which are being settling down right over here? Yes. Yes, it's that. But listen to me carefully. So basically they did their work. They broke down the organic matter. Now this part, this effluent which is there, it is pretty much safe to be released into river or any water streams out there because it is clean. Now whatever we have over here, this is super important. And where is this happening? In the settling down. So until now, I hope it's clear. We'll read and we'll come back, okay? So first one was this primary treatment, we studied it. Secondary treatment is biological treatment where you do aeration process. The primary effluent, that is the fluid, is passed into aeration tanks. Aerobic microbes form floc, that is bacteria plus fungal filaments. Microbes break down the organic matter, reducing the BOD. Yes, it breaks down the organic matter, and obviously when the organic matter is less, the BOD will be less because the aerobic bacteria will also get less. Now, biochemical oxygen demand. Okay, lower BOD means reduced pollution potential, whereas higher BOD means what? More potential out there. Got it? Now is the sedimentation process. So first is the aeration process that we did. The second process, sedimentation process, where what you're doing, you're taking the effluent is moved to a settling tank. Whatever effluent was there, we are taking it to what? The settling tank. The bacterial flux sediment forming what? Activated sludge. So now this flux which are sedimented out there, you have a name for it. You call this as a what? Activated sludge. So let's go back over here. Let's write that down. So this thing that is there, you call this as the activated sludge. And you know what? Some part of this activated sludge is put back into the aeration tank. For the next cycle to happen, for the next aeration process to happen, you need aerobic bacteria, right? So how do you get it? You have to provide it first, then they will be dividing it. So this acts like what? An inoculum, a base. Okay, so you have the activated sludge, some part of it you give back. Now what do you do? Anyway, this particular effluent has been all clear, you can put it in the river. Now what do you do with this part, the activated sludge part? You need to do something, right? What do? Basically, you transfer it inside the last thing, that is the anaerobic sludge digester. Anaerobic sludge digester. That means over here, no entry for oxygen. No entry for oxygen. There is no oxygen right up over here. What are you going to do? You are going to put this activated sludge over here. Why do you do that? Because this activated sludge which is there, it is going to be broken up, okay, by what? The anaerobic bacteria. Activated sludge are going to be broken by anaerobic bacteria, and basically it is going to release biogas. So basically from the sewage we got the fresh water that can be removed, and also we got what? Biogas that can be used in different households as well for production of electricity, for cooking purpose, anything. Always remember the biogas that we are getting over here is made up of three very important things. It's not just methane in the biogas. People always think it's just methane. No, biogas is made up of CH4, H2S and also CO2. So three gases are there: CH4, methane, H2S, hydrogen sulfide, and also CO2. Sometimes instead of H2S they also write hydrogen H2. It's totally fine, but please remember biogas is made up of these three things. What is this tank? This is anaerobic sludge digester, where you are taking the activated sludge from here and making it break down with the help of anaerobic bacteria. Here there is no entry for oxygen, then only it can produce biogas out there. The biogas consists of CH4, H2S as well as CO2. Got it? This is how it goes. If at all you want to take a screenshot, you can. Fine. So please remember where it is being formed. Settling tank is the place where activated sludge is being formed, and anaerobic sludge digester is where it is getting broken down. All this pieces of information is super important. We need to read it, right? So let's go ahead. Just a second. Come on. Yes. So effluent is moved, we read it, right? A portion of the activated sludge is sent back as inoculum. Now anaerobic digestion, remaining sludge is sent to the anaerobic sludge digester. Anaerobic bacteria digest the sludge, producing biogas, that is methane, CO2 and H2S. And finally the treated effluent is released into the rivers or the stream. So basically the effluent which is there, my dear children, can be finally released into any river and any places out there because it's super clean. Got it? Yes, ma'am. Fine. That's all. Settling tank, aeration tank, again settling tank, and then you have anaerobic sludge digester. Urbanization has increased sewage production. Yes, more urbanization is happening, more people are there, there is more sewage production. Insufficient sewage treatment plants lead to untreated sewage being discharged into rivers. And this can be very, very problematic. This causes pollution and spreads waterborne diseases for sure. Ganga Action Plan and Yamuna Action Plan aim to reduce river pollution. So if at all you are asked, what are the action plans that have been taken to reduce the river pollution, please know it is Ganga Action Plan and also Yamuna Action Plan. Ganga and Yamuna. It's a very famous two rivers out there, right? Ganga and Yamuna action plans are there. Okay. Now, the next thing that we have is biogas plant. Okay. So this is the biogas plant. Now, if at all you are a PU2 champion, you need to remember that you might be asked to make this diagram. Okay. So over here, always remember, what are you putting right here? The cow dung. The cow dung. Why are you doing that? Because to produce biogas, you have different ways. You just not have to rely on the sewage treatment plant. You can directly produce it. So with the cow dung, cow dung has methanobacterium. That is basically an anaerobic bacteria that can break down the things out there and producing the biogas. So methanobacterium is what? Anaerobic bacteria. Where is it found? In the cow dung. So that and water is being mixed, everything comes over here. This digester acts like an anaerobic sludge digester. So there is no oxygen up over here, and when there is no oxygen and you have anaerobic bacteria, my dear children, what can happen? Basically, you can see production of biogas. The biogas will be including CH4, CO2 as well as H2. These gases are being produced with the help of these gases are being transferred with the help of the chambers and is sent to different households. Now, whatever is left behind after the production is being sent to this particular area, that is a sludge. Okay, that is the area that has the sludge. What do you do with that? The sludge which is there being produced, you can use it as a fertilizer or as a manure. So all of it is being completely useful. Okay, so biogas is what? A fuel gas mixture, mainly methane, CO2 as well as H2. Methanogens, example methanobacterium, produce methane in anaerobic condition. What does it do? It produces the methane in anaerobic condition when there is no oxygen. Always remember, methanobacterium is an anaerobic bacteria, not aerobic. Found in sewage sludge and rumen of cattle, aiding in cellulose digestion. So this particular bacteria is actually there for the breakdown of cellulose. Cellulose, is found in the plants, and so cows eat plant, which is why they need something to break down the cellulose. Indirectly or directly, it helps, which is why it's up there in the rumen of the cattle. Now, development, how was it developed? It was developed by Indian Agricultural Research Institute, which is known as IARI, Indian Agricultural Research Institute. And apart from that, Khadi and Village Industries Commission, KVIC. So you have two names. This is an MCQ question that has been asked before. So biogas plant was developed by IARI, that is Indian Agricultural Research Institute, and we have KVIC, that is Khadi and Village Industries Commission. Two other things. Got it? Now, encouraged in villages for sustainable energy, visiting a biogas plant can provide practical learning. Yeah, that you know, right? Yes. So please remember this thing. IARI and KVIC, and please, while you remember this, also know what does all these things stand for. KVIC, Indian Agriculture Research Institute, and Khadi and Village Industries Commission. Got it? Yes. Next thing, you have biocontrol agents. Now we are ending up the chapter really soon, don't worry, okay? Two more things are there and then we are done. That is this and one more thing. So biocontrol agents, as in, you might have seen that you use pesticides for killing pests, right? In farmers, if at all you have seen, they use pesticides for killing pests. What if there are some organisms used to eat up these pests out there? That will be really nice, right? So these organisms which control the pests out there, you call them as biocontrol agents. And over here, you have the first thing, that is predatory insect. You have the first thing, that is what? Predatory insect. So ladybird beetle, my dear children, is such insect. What does it feed on? It feeds on aphids. It feeds on aphids. So ladybird beetle, which is red and black spotted beetles out there, what do? They feed on aphid, which are actually crop pest. Okay, apart from that, you have dragonflies. Dragonflies are very much common. They feed on mosquito larvae. Mosquito larvae is also a big problem for us. Yeah. So predatory insect is the first group where you have ladybird beetle, and apart from that, you have dragonfly. What does a ladybird beetle do? It is basically feeding on aphids. What does dragonfly do? They feed on the mosquito larva. Done. Next is microbial biocontrol agent. So predatory insect is done. This been done. Now we talk about microbial organisms that are going to feed on the insects. Let's see, or basically kill the insects, not feed on the insect, kill the insects. The first one is Bacillus thuringiensis, a very, very famous guy. How does it work? Bacillus thuringiensis is a bacteria, my dear children, which has some protoxins within it. That means some toxins that has an ability to kill certain group of organisms. But for that, it needs correct environment. So suppose this particular bacterium is eaten by a caterpillar. What happens? The bacteria goes to the mid-gut region, right? It reaches the mid-gut region. In the mid-gut region, when this bacteria reaches, the protoxins, my dear children, will be converting, just a second, will be converting into active toxins. And when they convert into active toxins, they can cause perforation. When they cause perforation, what is going to happen? Basically, this organism is going to die. Why do you think this happened? Because in the mid-gut region, my dear children, of the organism, you have an alkaline pH. What do you have? You have an alkaline pH, and at this alkaline pH, the protoxins converts into active form of toxins, and that is how it will be able to do its work. So that is how the killing happens, okay? So microbial biocontrol agent, the first one is Bacillus thuringiensis, a bacterium whose spores kill butterfly caterpillars when eaten. So available as dried spores. Now how it is being taken, this is also an important question. It is not fresh spores that you take it. So I saw an MCQ question where they mentioned every single thing, just like this, except that instead of putting dried, they put fresh ones. So please understand, it is not fresh, it is dried ones, okay? Available as dried spores in sachets, mixed with water and sprayed on the plants like brassicas, that is cabbage and cauliflower, and also fruit trees. So what do they do? You have the dried spores of this particular organism, you mix it with water and then spray it. On what? On brassicas such as cabbage and cauliflower, and also on the fruit trees. Got it? In the caterpillar's gut, the Bt toxin is released, killing only the caterpillar without harming the other insects out there. So it is very insect specific, you should know that. So it is Bt, that is Bacillus thuringiensis, which is the first example of microbial biocontrol agent. Predatory insects you studied, this is the second one, okay? Next you have fungal biocontrol agent. So there it was a bacteria that was acting like this. Here you're talking about a fungal one. So in fungal you have trichoderma fungi. Remember trichoderma polysporum, which was producing what? Cyclosporin A, that is an immunosuppressive agent. Remember, if at all you're not able to recall, it's okay. Okay, it's okay, don't worry. You will get a hang of it. So trichoderma fungi, this is something else. Over here, this is a free living fungi found in the root ecosystem. This as in when I said it is different, I meant that it is doing a different function all over here, totally different, and I have to check the species also. Anyways, so forget that. It's a free living fungi found in the root ecosystem. It is found in the roots of the plants, and it is effective in controlling the plant pathogen, that is the disease causing microbes. So basically I'll tell you, there are some funguses which goes inside the root and cause trouble. To kill those funguses, you have another fungus, and that is trichoderma fungi. This fungi is going to act like a biocontrol agent because of the same reason. Okay. Apart from that, the last one that you have is the viral biocontrol agent, that is the viruses. And the first group of viruses is baculoviruses. They come under nucleopolyhedrovirus. Baculovirus, they come under nucleopolyhedrovirus, and they infect only specific insect pests and arthropods. Arthropods as in basically insects and things like that, okay? So infect only specific insect pests and specific arthropods as well. So insects are coming on the arthropods. It means that apart from insects also, there are other things also which are being harmed with the help of baculoviruses. Okay, so virus biocontrol agent, you have example as baculoviruses. They come under nucleopolyhedrovirus. This is also a question, in which category they come under. So it is nucleopolyhedrovirus, and they are insect group specific. They kill arthropods. Okay. Now they are eco-friendly, does not harm plants, mammals, birds, fish, or beneficial insects. Useful in integrated pest management and in ecological sensitive areas. So all of them are super important in areas where it is super sensitive. You cannot put in any organism out there, right? So these are sensitive areas which can cause really bad damage if at all there is some problem. So in these type of areas, these organisms are being used, and it can be used in integrated pest management also. Okay. So that's the general thing out there. So what is the name of the things that you need to remember? Starting with the first thing, let's recall, okay? Let's recall. The first one that we have is predatory insects. First is ladybird that kills the aphids. Second one is dragonfly that kills the mosquito larvae. Now, we're talking about bacterial biocontrol agent. The first one is Bacillus thuringiensis. It kills what? The caterpillars, etc., etc., with the help of toxin production. Fine. How it is being put? Basically with the help of dried spores, we mix it with water and spray it on brassicas and also on fruit trees. Okay. Now the next thing that we have is what? Is fungal biocontrol agent, and the first one is trichoderma. It is a root fungi. It's a free-living fungi actually. It does not depend on the plant for any sort of food. It is a free-living fungi. It can sustain, but it protects the plant from other fungus which are there in the roots. Okay, now we have viral biocontrol agent, and the first example that you have, first and the last, is baculoviruses. They come under nucleopolyhedrovirus. They are also insect group specific. They kill variety of arthropods as well. So insects come under arthropods. Apart from that, there are other organisms also coming under arthropoda. So they also would be killed with the help of these viruses out there. Got it? Yes, that's all. So just give me a second, okay? Yes, so it's the same Trichoderma polysporum which produces cyclosporin. I just meant to confirm that. Okay. Now the last part of this chapter is biofertilizers. Similarly, we can use fertilizers which have some chemicals in it, but obviously these chemicals can cause imbalance in the soil, okay, pH imbalance in the soil, which cannot be really good for the other plants, which is why we need to be really conscious. We can probably use biofertilizers instead, which do not have these side effects, right? So the first group of organism is nitrogen fixing bacteria. What do you mean by that? These organisms provide nitrogen to the soil, or basically to the plants. And obviously, when you provide nitrogen, so it's like a fertilizer, it's acting like a fertilizer only, but an organism is doing it from its own end. Example for that is rhizobium. So rhizobium forms root nodules in the legumes, fixes atmospheric nitrogen into usable forms. So atmospheric nitrogen is in the form of N3, that means, just a second. Atmospheric nitrogen, we say that suppose there are two nitrogen molecules, they have triple bond between them, which is why it cannot be broken down very easily and cannot be taken by plants so easily. You need someone to break it down, to convert into something else, such that the plant can really take it up very easily. And rhizobium does that, which is why it is said that it fixes atmospheric nitrogen. By that it means that it converts the atmospheric nitrogen in some other forms which can be useful for the plants. And they are found in the root nodules in the case of leguminous plants. For example, pea plant and things like that. Now we have azospirillum and azotobacter. Acetobacter is different, okay? Acetobacter aceti is for acetic acid. This is azotobacter and azospirillum. Both of them are starting with azo. They are free-living bacteria that can enrich soil nitrogen content. So they are not like rhizobium. Rhizobium actually basically is a symbiotic association. They live together. It's not free-living. Rhizobium is not free-living. But azospirillum and azotobacter, they are free-living. They're independent of the plant, but they still they increase the soil nitrogen content out there. That is why these two things are different. One is basically not independent, one is dependent, whereas the other two are somewhere saying free-living. Okay, somewhere free living. They are not exactly living because of the plants out there. Got it? Yes. So these are what? The bacteria. Now over here, I will come directly to here because this is a common doubt that you guys have. Cyanobacteria is kept separately. Cyanobacteria is also a group of bacteria, but they do, my dear children, they are responsible for producing their own food. They are the ones that produce their own food. Okay, that is why probably they are being kept differently. Example is Anabana, Nostoc and Oscillatoria. What do? They also fix atmospheric nitrogen and increases the soil fertility. So when you are asked about plain bacteria, go with this. When you are asked about cyanobacteria, this blue-green algae, please go with this. Okay, they are autotrophic in nature. What do? What are the examples? Anabana, Nostoc and Oscillatoria. So it is no, I am not coming to play. So no. Okay, so fix atmospheric nitrogen, increasing the soil fertility. It is used as biofertilizers and also add organic matter to the soil. This is important. It is used as biofertilizers where? In the paddy fields. And also adds organic matter to the soil. Know that. Okay. So rhizobium is not free living. It forms root nodules. In the case of legumes, it fixes nitrogen, that is there. Azotobacter and azospirillum, they are free-living bacteria which increases the soil nitrogen content. Cyanobacterium is autotrophic bacterium, okay, which prepares its own food. And over here, Anabaena, Nostoc and Oscillatoria are examples for that, and they fix nitrogen. And apart from that, basically they're really used as biofertilizers in the case of paddy fields, okay, and they also provides what? Organic matter. Now apart from that, you have mycorrhizal fungi, okay, mycorrhizal fungi, that is fungal biofertilizers. Here fungus is going to play a very important role. So listen to me. This fungus which is there, so I have the roots of the plant, this is the roots. The roots and the fungi, my dear children, they are having a symbiotic association. They are helping each other. They are going to help each other. They are going to remain together. Okay. This fungus which is there, it belongs to glomus. It belongs to glomus. Now the question over here that is usually being asked is that, what does this fungus do? This fungus also have root-like structures which can go seep into the soil and bring back very important nutrients. For example, what? Phosphorus. For example, phosphorus. So phosphorus is provided by the fungus to the plants. Apart from that, it basically helps provide resistance. Okay, to what? To pathogen out there. To pathogen. Apart from that, tolerance to salinity. Resistance to pathogen. Apart from that, tolerance. It can tolerate the soil which has high amount of soil, so what you call salt content, that is what you call a salinity. Apart from that, if at all suppose this area does not have water, it will help the plant survive because it will make the plant tolerant to drought condition also. And my dear children, last but not the least, one more thing, that is overall plant growth and development. Okay, overall plant growth and development can also be helped with the help of this fungus. So glomus mycorrhiza forms a symbiotic relationship with the plant root. So listen to me. This association between the roots and the fungus is known as mycorrhiza. The fungus belongs to what? Glomus. As I told, it absorbs phosphorus from the soil and passes it to the plants. Apart from that, provides resistance to pathogens. Apart from that, provides tolerance towards salinity and drought. Apart from that, helps in overall plant growth and development. What does the plant do? Plant is responsible for the process of photosynthesis, can prepare the food by itself and pass the organic matter to basically the fungi. So fungi benefits by receiving organic nutrients from the plant because the plant anyway will be doing photosynthesis. So that is why I said that they have a relationship between them. Okay, a symbiotic relationship between them. Always remember, these are the only things that are provided by the fungi. If at all you are asked, and it is being asked also, does the fungus helps in increasing the yield of the plant? That means production of the plant? The answer is no. It has nothing to do with the yield of the plant. Overall plant growth and development is there, but it has nothing to do with the yield of the plant. Please remember that. This is super, super important. Just give me a second. Someone has come. Just a second, children. Okay. So that's all. So can we revise once? Okay. So nitrogen fixing bacteria, we have normal bacteria as in heterotrophic bacteria. Rhizobium, which is forming the root nodules in the legumes. It fixes nitrogen. Apart from that, you have azospirillum and azotobacter. Done? Done. And azospirillum and azotobacter are free living. Please remember the terms. These are important. You can be asked in any form. Now, mycorrhizal fungus, fungal biofertilizers are there. And here the fungus belongs to the glomus community. And my dear children, remember they help in the absorption of phosphorus. That is the first and the foremost thing. Second thing, resistance to resistance to pathogens. Third thing, tolerance to salinity and drought. Fourth thing, what? Fourth thing is this thing, that is overall plant growth and development is being handled by this particular organism. Now what do you have? You have the fungus. Yeah, that's all. And now the plant which is there, it provides the fungus what? The organic nutrients because it can do photosynthesis. Now we have cyanobacteria. Cyanobacteria are autotrophic bacteria out there. They prepare their own food. Apart from that, they help in providing nitrogen also. For example, Anabaena ANO, right? Anabina, nostoc, as well as oscillatoria. Fine. And they are acting as biofertilizers, especially in the paddy fields. And my dear children, they also add organic matter to the soil. These are the only things that you need to remember from biofertilizers part. And with this, we end the whole chapter. So I obviously think that there are chances that we might forget things. So what do we need to do? We need to go back. Okay, we need to go back, just go through each and everything and just try to revise and write. Just try to revise and write and do this every single day. The time and all will be, will go lesser. And by that, after some point of time, you just have to look at it. But if at all you're thinking that, okay, today I studied, tomorrow I'll just look at it and I'll just remember like that, it won't work. You have to sit and write. Okay, you would have to sit and write. After a few days, you can rely on reading only, because by that time, our mind might have stored a lot of things out there. Okay, so please do that, and please let me know if at all this video was helpful for you. And my dear children, if you have any doubts that is coming into your head, do let me know about that also. Wishing you all the best. Please take care of yourselves. Bye-bye. And remember, Methanobacterium is an anaerobic bacteria. Bye-bye. Take care.